evening, everybody, and welcome to today's NanoTalk on the Physics and Chemistry Nobel Prizes. I'm Ophelie, and this is Lou.
Hi, I'm Lou.
And we will be your hosts for the evening. As some of you might not be completely familiar with this event, let me give you a quick overview of what will happen. For every NanoTalk, we invite two young scientists to give a 15-minute talk about their research field. After both presentations, you have the opportunity to ask questions. The NanoTalks have several aims. On one hand, the format offers a training platform for speakers to help them improve in the domain of science communication, and of course, on the other hand, the audience gets to know a little more about current research and new developments. The NanoTalks also help facilitating networking between audience, speakers, and organizers. Before Lou tells you more about the role and aim of hosts and organizers, let me show you how to get the most of the NanoTalk by asking a question on the anonymous forum. Our experts will be answering these questions today at the end of the evening. The platform is called Sly.do, and the code today is nano-nov.
Now I'm going to introduce to you the organizer of this event, the organization REACH. REACH stands for Research, Think and Change, and is an independent think tank for a science-friendly culture. It promotes the dialogue between scientists and the public. REACH organizes events that inform about relevant topics in science, more specifically in the fields digitalization, biomedicine and health, and energy, climate, and environment. REACH also publishes blog posts on its website and gives out policy recommendations. There are over 250 volunteers working for REACH in Zurich, Bern, Basel, and Lausanne. A popular program of REACH is SIMPACT. Ophelie and I actually are SIMPACT fellows. SIMPACT enables young researchers to have the opportunity to organize events such as the NanoTalks. The aim of SIMPACT is to make those who are interested in science aware of their responsibility and to enable them to make a difference with their knowledge. SIMPACT encourages critical reflection, communication, and problem solving. I'd like to remind you that questions tonight can be asked on the platform Slido with the code nano-nov. You can also look it up in the description of the video. The first speaker tonight will be Kenny Jungfer, and he will introduce to you CRISPR-Cas, the revolution in genetic engineering. Kenny is a PhD candidate who works in the lab of Martin Jinek. Funnily, Martin Jinek was the first author of the seminal CRISPR paper and worked in the lab of Jennifer Doudna, so of one of the women who won the Nobel Prize in chemistry. So this is kind of a link between Kenny and CRISPR. Another link is that Kenny could actually directly profit from CRISPR, or more specifically, his plants could profit, because even though he's a biologist, he's not very good at keeping plants, so he said. And now I'll pass the word to you, Kenny.
Lou, thank you so much for the kind introduction. I will share my screen if it works. Do you see my screen? So, yeah, thanks again for the kind introduction. And as Lou already told you, I'm today explaining you a bit about CRISPR-Cas and its impact on today's world with the title, A Revolution in Genetic Engineering. So in recent years, CRISPR-Cas very much dominates the scientific media landscape, with bringing up headlines like CRISPR-Cas9, the gene editing tool that has revolutionized molecular biology, starting controversies like the CRISPR baby scandal, what's next for human gene editing, but also giving glimmers of hope, like CRISPR takes its first steps in editing genes to fight cancer. And these are only some of the headlines that can be collected in order to explain how groundbreaking CRISPR actually is. Not to forget, of course, the Nobel Prize in Chemistry that was awarded to Emmanuelle Charpentier and Jennifer Doudna in October 2020. And my aim today is kind of to bring you CRISPR-Cas a bit closer, tell you what its function is, and also explain to you why it's such a groundbreaking biotechnology. And in order to do that, I want to talk about a genetic disease that where a lot of progress was made in the last few months. And this is about hemoglobin disorders. So normally, red blood cells are the vehicles of oxygen within our bodies, having hemoglobin as the molecule that binds the oxygen and releases it at the organs that require the oxygen, actually. So you can think about hemoglobin in a way like a mailman who is picking up the packages at the reception point, like the lungs, and delivering it or driving in the post vehicle, the red blood cells, and delivering it to the organs, or delivering it to the customer, who then are the organs receiving the oxygen. Unfortunately, there are some persons who have a mutation in the gene encoding for the beta subunit of this hemoglobin, the gene called beta-globin. And this mutation causes the abolishing of the production of beta hemoglobin, therefore leading to a defective form of hemoglobin that only contains the alpha subunits of its molecular structure. And this reduces the efficiency of oxygen binding, and it also leads to a strong deformation of the red blood cells. And you can imagine, staying with the example from previously, you can imagine it like the mailman, for example, is catching the coronavirus and can't work anymore. So someone else has to be exchanged for someone else who is now taking over the job, which is unfortunately someone from the office who doesn't know how to drive the van, and he also doesn't know the fastest way to go to the customer. And this is actually taking place with the hemoglobin. The oxygen binding is reduced, and from time to time, there are crashes that lead to severe symptoms. And these severe symptoms include organ dysfunctions like jaundice or cirrhosis, but they can also lead to a strong expansion of the bone marrow that eventually leads in the most severe form of a disease called beta thalassemia to the deformation of the head of the patients. So around 7% of the world's population are actually carriers of mutations in genes that could cause hemoglobin disorders, and about 300,000 to 500,000 babies born every year are actually generating these symptoms that have to be treated. And the treatment looks as following. They are dependent of lifelong blood transfusions. Those are connected to very painful iron chelation therapies, but they also need regular bone marrow transplants in order to keep the healthy cells dominating this system. So this is a lifelong treatment that is only tackling the symptoms, but doesn't pose an actual cure to the sickness. And kind of driven by the severity of these hemoglobin disorders, scientists were trying to find a solution in order to relieve the patients and make them independent from these blood transfusions and bone marrow transplants. And in their search, they were kind of successful in the last few months. And on their search, they were inspired by nature, and to be more precise, by a bacterium called Streptococcus pyogenes, which ironically is a human pathogen that induces hemolysis, meaning that it destructs red blood cells. But this bacterium evolved a system, an immune system, that defends it from bacteriophages. And this defense system is called CRISPR-Cas. CRISPR being short for clustered, regularly interspaced short palindromic repeats. And probably from that name, you already know why we use the more crisp sounding name CRISPR. And this system works as following. So bacterial cells are constantly under attack by small viruses called bacteriophages. And these bacteriophages attach to the bacterial cell and inject their DNA harboring the replication machinery of the bacterium in order to replicate itself. That eventually leads to the death of the bacteria itself. But to defend itself, the genome of the bacterium contains the so-called CRISPR array, which is, you can imagine it kind of like a memory of previous viral attacks. And this memory works as following. The viral DNA gets recognized by the system and gets inserted into the CRISPR array. So basically being an exact copy of the viral thread that is now in the genome and can be used in order to defend the system, and saving it when the virus is attacking again. So now when there is another viral attack, the CRISPR array can actually be, or the information saved in the CRISPR array can be translated to a guide that is identical to the DNA that was inserted by the bacteriophage, and called the CRISPR RNA. And this CRISPR RNA is bound by Cas9, the molecular scissor from the CRISPR system. This Cas9 scissor gets cocked and triggered by a trans-activating RNA called tracer RNA. And together, the Cas9 complex is recognizing the DNA of the virus in a sequence-specific manner. And now to rewind a bit, because most of you or some of you might have forgotten about their biology lessons and previously. So the genetic code is basically composed of 4 basic units called the bases. We have the bases A, T, G, and C, where A is binding to T and G is binding to C. And and having those bases in series generates a unique fingerprint for the specific organism called the genetic code. And this genetic code is very specific for the different organisms. And now we have the CRISPR RNA carrying an exact copy of the viral DNA. So what happens now is that the region that is identical to the viral DNA is binding one of the strands, allowing Cas9 to access both of the strands, cleaving those strands and destroying the DNA, and therefore defending the bacterial cell from the viral threat. In 2012, Jungfer, Doudna, Emmanuelle Charpentier made their groundbreaking work on CRISPR-Cas, together with the contribution of my current supervisor, Martin Nienek, as Lou already told you. And what they found out was that a specific region in the CRISPR RNA called the seed sequence is essential in order to provide a sequence-specific cleavage of the target DNA. And this seed sequence is again the copy of the DNA from the virus. Moreover, what they achieved was to combine the CRISPR RNA and the tracer RNA to form the single guide RNA that can then be used as a very easily modifiable tool that is not only highly specific, but also very versatile, because you can choose any seed sequence you want using molecular biology techniques. And this, in the end, is the main point for receiving the Nobel Prize in 2020. So how can you now use CRISPR-Cas to do gene editing? So you have in the first step, you have to design your CRISPR, the single guide RNA, and specifically you have to design the CRISPR, the seed region, in a way that it specifically targets the DNA that you want to cleave. Then Cas9 is bound to the target DNA. It opens the 2 strands. The seed regions can form complementary base pairs, and this induces the cleavage, so the cut of the DNA, leading to 2 loose ends. So now we have the cut DNA, but however, we still don't have any genes edited. This is where the DNA repair machinery of the bacterial cell kicks in. There are basically two mechanisms that can be used to edit genes. One is based on homology-directed repair, or short HDR, where you supply a sequence, a random sequence that you designed in a way that you want the sequence to be, together with two regions that are homologous to the sequences surrounding the gap in the DNA. The host repair machinery is now taking this homologous DNA and inserts and fills the gap that was left by Cas9 by inserting the DNA that you provided. And with that, the scientists can actually rewrite the genetic code of the organism. So scientists at Harvard University are now trying to use this method in order to resurrect extinct species. And the most prominent example of that is the woolly mammoth. And what they do is taking the DNA of frozen mammoth DNA and combining the sequences with Asian elephant DNA, creating a mixture of both that equals the DNA of the mammoth. This can be taken and reintroduced into a reprogrammed skin cells that now have the function of stem cells, so they can replicate themselves and form the embryo that then grows in an artificial womb that eventually leads to the resurrection of this woolly fellow. But now back to the problem of the beginning, the genetic disease. And for that, there's another mechanism that can be used in order to, or that was used in order to tackle the hemoglobin disorders. And this is called non-homologous end joining. And in this case, you don't provide a DNA, but the host DNA repair machinery is actually trying to fix the DNA double-strand break itself. However, this mechanism is highly error-prone, meaning that it often leads to mistakes when repairing the two DNA strands, meaning that bases can be added, exchanged, or also deleted from that. And this you can imagine as a type of shut-off button that leads to a knockout of the gene. So the information encoded by the gene cannot be read properly anymore, and therefore being silenced. This was also used, or just to give you an overview, these gene knockouts have a great implication for basic research, as they give us the opportunity to analyze the function of different genes in an organism. By knocking them out, we can actually observe the effects of the gene knockout on the entire organism, and then we can conclude what the function of the gene is in this organism. But this is also the mechanism underlying the treatment of the hemoglobin disorders. In 2019, Vertex approved the first drug, CTX-001, that is based on gene editing for phase 1-2 clinical trials. And this was a huge win for recreational medicine. And this therapeutic works as following. So in the patient's cell, in the patient's bone marrow, you have the hematopoietic stem cells that generate the red and white blood cells. And these are extracted from the patients and then targeted for gene editing. And what do they do? In those hematopoietic stem cells, there's a gene called BCL11A. This encodes a repressor of fetal hemoglobin expression, meaning that it inhibits the formation of this fetal hemoglobin, short HbF. Now the idea is to use CRISPR-Cas9 to knock this gene encoding for this repressor in order to re-achieve the production of this fetal hemoglobin that then eventually leads to the formation of normal blood cells. These are then reinserted into the patient's bone marrow, where they then boost the production of healthy blood cells that should exchange the defective blood cells. You can, going back to the example of the beginning, it's like the office mailman that was substituting the sick mailman gets now exchanged by a mailman that was retired before, but already knows the job. He knows how to drive the vehicle. He knows how to drive, where to drive, and how to supply the customer with the packages. And this is the fetal hemoglobin in this case. And extremely interesting, 2 months ago, they published the first results on this therapy, indicating that after 5 to 15 months, 50% of the fetal hemoglobin exchanged the defective hemoglobin in those patients, making all 3 patients independent of blood transfusions. And this is a great leap for recreational medicine again. Even though it's only based on preclinical or early stage clinical trials, it's still a big prospect for the future of CRISPR-Cas and its usage in medicine. So to briefly sum up what I was talking about, so I gave you an example of the importance of CRISPR-Cas in basic research, where I just briefly mentioned that we can edit genes or knock out genes and analyze their function then in the specific organism. I also told you about the de-extinction of organisms like the woolly mammoth, but also there are different other opportunities. Then we can also use CRISPR-Cas in medicine in order to treat genetic diseases that until now were not curable. But there are many more examples of the application of CRISPR-Cas. For example, tackling the challenges surrounding COVID-19, where we can use CRISPR as a kind of fast detection system in order to make all of our measurements more efficient. There's also a big influence in agriculture, where gene editing is a long used method in order to change crops in a way that they produce their fruits in a more efficient way. And this is also one of the aspects that will bring the importance of CRISPR much further. But then one thing that I unfortunately didn't have time enough to talk about is also the ethical debate. Should we even use CRISPR to do all these things? And I can assure you that behind all of those things, there's a long debate in science to talk about the aspects, the benefits, and also the negative aspects of the CRISPR-Cas usage in editing the respective genes. Unfortunately, I wasn't able to talk about it, but I'm happy to answer your question regarding that in the Q&As later on. If some of you got more excited about CRISPR now, here's a list of very interesting links that lead you to the different aspects of CRISPR that I presented to you now. With that, I thank you very much for listening, and feel free to ask your questions later on.
Thank you so much for your talk, Kenny. Let me remember the audience to ask questions on Slido. Kenny will be answering them at the end of the evening. Now, let me introduce you to our second speaker of the evening, Raymond Angeli. Just like Einstein, Ray did his master's at ETH Zurich and his PhD at the University of Zurich. Today, Ray works at Credit Suisse as a quant. Ray isn't just passionate about physics, but he also enjoys languages. Indeed, Ray studied a lot of languages next to his PhD and his studies in general. In the next few minutes, you will get an insight on gravity. Ray, the floor is yours.
Okay, is that better? All good? Okay, so I start again then. So we're looking at the very large telescopes in Chile. The telescopes are pointed here into the center of the galaxy. Now, our galaxy is it's a thin disk, and the Earth is located about halfway towards the edge on one of the spiral arms. And so by looking into the center, we see this disk, this Milky Way in the sky. Now, if you zoom in a little bit, and with a bit of imagination, you'll be able to see the constellations of Sagittarius and Scorpio. And the galactic center is located about, somewhere in between these two, although I've taken a bit of artistic license in placing them exactly where they are. Now, if you look even closer, right, and even closer, we are going to see some interesting observations. So this is a series of images, quite raw, taken by the Keck telescope in Hawaii. And you can see some orbits. What you're seeing are stars right in the center of our galaxy. And these stars are orbiting something, right? This is an extremely interesting system to study. And I'm going to go to the next slide, which is going to take us a little bit away from the rawness of this image into something slightly more reconstructed, slightly cleaner, but still directly from these observations. This is work that was done over a period of about 30 years by two teams, the American team using a telescope in Hawaii and the German team using a telescope in Chile. These stars are orbiting something, and these stars are orbiting something extremely fast. In fact, some of them get up to a few % of the speed of light while they're doing this. So the gravitational field is extremely strong. And their orbits, although they look like ellipses, they're not quite ellipses. We understand Newtonian gravitational theory extremely well for a very long time, and it's been able to explain almost all gravitational phenomena, but something is wrong with it, and that theory does not work when we look at this picture. So not only are these orbits actually in very strange shapes, but they're orbiting something that we can't see, something that's black in the center there. We don't know what that is. Is it a collapsed star? Is it some kind of neutron star that emits no light? Is it a form of dark matter? Is it something that we haven't even thought of before? On the top left, you see Andrea Ghez. In the middle, it's Reinhard Genzel, and Roger Penrose in the top right. Andrea's instrumental telescope, you see in the bottom left, that's the Keck telescope in Hawaii. Reinhard's, you see in the bottom. And Roger Penrose, who I've not really mentioned yet, he's a theorist, which means unlike the other two who are observers, Roger's tools are mathematics and I guess a couple of doses of imagination. So Roger really brought our understanding of the geometry of space-time under extremely large gravitational fields into perspective. And he helped us understand and developed a lot of the mathematics, which help us understand how objects move in such space-times. Now, if you apply some very, very basic ideas to the orbits of the stars that I show you, you learn that you will see that in the center, the only way that those stars could be moving so fast around this common center is if something was there that we don't see, right? Something was there that has a mass of about 6 million times the mass of our sun. So it's something extraordinarily massive and powerful, and yet we don't see it, right? We're going to take a bit of a different perspective. So we take a step back now, 100 years or so. This is a young Einstein. You don't usually see Albert Einstein so young in images. And I took it because I guess this is about how old he would have been when he started thinking about the general theory of relativity, the new description to how gravity works. Einstein has many, many achievements, and I certainly think he discovered so much, and certainly his most profound work was in theory of gravitation. Einstein looked at Newtonian theory and he thought, this just doesn't make sense. It's going to break down, and he found so many inconsistencies, and he replaced the theory of general relativity, of Newtonian gravity, with the theory of general relativity, a new theory of gravitation. There are a lot of things, a lot of consequences out of this equation. And on the left-hand side, you see these R's and the G's, right? On the right-hand side, you see the T. It's deceptively simple like this. There are just a few letters here, right? But there are 10 equations in there, and if you expand them further, you'll fill many, many, many pages just with the equation before you even start to work with it. They're very difficult to solve. But they work very well in describing early universe cosmology, describing the formation of the universe. Everything that's got to do with gravity is very well explained by this equation. In the 1960s, a mathematician physicist called Schwarzschild, he found a unique solution to this equation, which was considered a mathematical fantasy at the time, something that we cannot have in reality. And like if we look again at these equations on the left hand side, the R's and the G's, they encode the curvature of space-time. And on the right hand side, you see this T mu nu. This is the energy momentum of space-time. And what it says here, this equation, is that the energy momentum of space-time curves space-time. And in turn, the curved space-time, which is a gravity, really tells energy and matter and momentum how to move. So that's very key. I'm going to say it again. Energy momentum curves space-time, and curved space-time tells energy and momentum and mass how to move. Now, the unique solution that I was referring to earlier to these equations from Schwarzschild made no sense at the time. He found a solution in which a spherical region of space had a gravitational field so powerful that no light could escape. It didn't really make sense to people. We don't see anything like this. It's probably just a mathematical artifact, whatever. A lot of theoretical work was done on that. It was a very interesting space-time to discover, to work with, at least from the mathematical perspective. But yeah, and I mean, this thing is called a black hole, right? What I'm talking about, you might have heard of it. So the solution of a black hole region of space-time that's so powerful that no light can escape. I've put an image of it on this slide, right? I'm being a bit facetious now because that's exactly it below the equation, right? Is nothing. Right? When you look towards the center of the galaxy, the common center around all of these stars, you also see nothing, right? I've taken a bit of artistic license here. This is a computer-generated image of the space-time surrounding a black hole with an accretion disk where you have a lot of very, very hot matter getting sucked into this region. And the silhouette that this matter makes as it radiates away its energy, you can see in the picture, so that the dark center is the black hole. But if you look into the galactic center, right, where all the stars are, you see nothing. Now, obviously, you know what I'm getting at. Through these observations by these scientists for many years, right, 30 years, studying the shape of these orbits, studying the frequency of the light, the color of the light that arrives in our telescopes of these stars as they approach this extremely strong gravitational field in the galactic center. This has unequivocally now culminated in pretty much perfect, extremely strong evidence for the correctness of these equations. And before this, we had many reasons to doubt that this fundamental theory of gravity is ultimately correct. And there are other theories of gravity that are even more complicated than this, and it turns out they're all wrong, right? And this is the one that works. It's written down 100 years ago, and it's the work of these scientists, as well as perhaps a few other discoveries in the past years that have really solidified this into correctness. I'm going to keep it local, right? Einstein worked in Zurich, German origin. So he worked in Zurich for a while. But we're going to jump forward 100 years to 10 years ago. So University of Zurich was involved in part of this work. So I work with Prasenjit Saha. I'm a professor at the University of Zurich. And we are theorists. We don't know how telescopes work very well. We worked on the orbits of these stars, and we studied them from a theoretical perspective. We computed the consequences of the general theory of relativity and other theories of gravity on this system. And in particular, we worked on how light travels in this environment of extremely strong gravity around a black hole. What you see here is a very dirty simulation that we put together, I guess, a few months into my PhD. I apologize, it's not as sexy as the images showed earlier, but this is the way theorists work. It was hacked together at some point. You seeing a star, the blue track orbiting this black hole, the red lights are photons, little bits, rays of light that are getting absorbed by a telescope on what we see as the Earth far away there. Now, this star has been brought really close in. You can see that the orbit is not an ellipse, and you can see, maybe you can see right now, that those rays of light are not exactly straight. Right? They're going to get contorted and lensed as they go around the black hole. And you're going to see it happen when it approaches the black hole. So it's going to, as the gravitational field gets stronger and stronger, so this is a consequence of a bunch of mathematics, some computation, and bam, there it happens. And that signal is something very particular to general relativity, right? So this is my last slide. Just a few closing points, a few final thoughts. When we worked in this field, we didn't expect that it would culminate in a Nobel Prize. Because the Nobel Prize in physics is usually given to scientists who manage to discover something, which then discovers some fundamental physical principle, which then turns into really strong benefits to humans and society. For example, the LED, right? The fundamental principles which culminated in the LED and allowing you to go to the bathroom in the middle of the night without tripping over something. They got the Nobel Prize a few years ago in physics. The original work that resulted in us being able to develop the technology for MRI machines, the discoveries of those physical principles, which has ultimately led to saving countless lives through medical diagnostic techniques. These are the people who usually get the Nobel Prize. Why do we do this? There's no application here, right? At least not yet. And this is my point is that we just do science because we can, because it's fun and it's interesting, and we don't have an application for it yet, but that they award the Nobel Prize to a group of people without yet having a strong application. That's great. It's been done before for the Nobel Prize in Physics, but it's not often that it's like that. Who knows, perhaps in many years in the future, before humanity doesn't manage to destroy itself before that, we'll find a way to use this and turn it into technology. How can we build from strong gravitational physics in the neighborhood of a black hole? That might be exciting. So we're very proud and very happy that this was being awarded to those three scientists. It's absolutely amazing, and of course we're proud to have been involved in a very modest, small way here in Zurich. So yeah, this animation plays on awfully. I don't know, Joy, I'll give you back the floor.
So thank you very much, Kenny and Ray, for your interesting talks. We've now arrived at the Q&A session of this talk. And I would ask a first question to Kenny. One of our watchers has asked on Slido, when can we first expect the first CRISPR-based therapeutics to be available to common people?
And that's kind of difficult to answer because it always depends on the results of the early stage clinical trials, right? So it's 1.5 years approximately in those clinical trials, and to expect it to be available before the next 10 years would be kind of dreaming a bit. So there are long-term studies, for example, that are estimated to be finished in 2039, so quite long from here. But hopefully, at least in the US, the drug has the potential to be approved in the fast track, and this could shorten the time. But I think in the next 10 years is, I mean, it's such a completely new technology that we have to get to know all the details before that.
And so I expect in other fields of application, CRISPR could be implemented much faster, like for example, in plant technology.
Yeah, I mean, this is already taking place, right? So not only in basic science where we use CRISPR routinely, basically, we also generated already crops that are, for example, resistant to a specific threat. Like there's a citrus canker, for example, that is a disease of citrus plants that reduces the yield of fruits dramatically every year. And there's a genetically modified variant of the citrus tree that has a mutation or knockout in a gene that helps the plant to resist this bacterium, the Xanthomonas axonopodis. And yeah, these are things that already work out, and this is actually a cool option to also think about changing from using just changing our use of herbicides, etc., to get to more sustainable solutions.
Thank you so much for your answer, Kenny. Maybe now we'll go more into some physics, and I'm going to ask a first question. How do experimental physicists and theoretical physicists work together on such topics from like what happens in space and black holes, etc.?
That's a funny question. It's a great question, because sometimes we don't work together, and sometimes that's the problem. We all want to know it all, right? If only we could be all theorists and all experimentalists, but amount of knowledge that you have to know of both is too much for the time we have. The dynamic, at least in the field that I've been exposed to, is really that we take physical principles as a theorist, you compute the consequences, you come up with ideas, and you come up with, hey, maybe that's real. And then the scientific community, experimental community can take it up, and they can check for it, they can test, they can say, no, that's bullshit. Yes, that's not that bad. Maybe it works. But interestingly, it also happens the other way around, right? In the topic that we just spoke about, it was what I just said, right? It's theory first. Experimentalists find something that, hey, could look like that. But often it's the other way around. Sometimes the astronomers, they point the telescope at the sky and they're like, what the hell is this, right? And then the theorists come in and they go, oh, it's that, and then they do more observations and they go, no, it can't be, right? And then it works like that, right? And eventually the theorists manage to explain it, hopefully, right? Usually that's how it works, back and forward. That doesn't really describe the work together, but it describes the two roles regarding the actual Zusammenarbeit, the way they communicate with each other. Sometimes some broken communication because we speak different languages, but good experimental teams will have some connections with a few theorists, and good theorists will have some connections with the experimentalists.
So do you think that the research could be more efficient if theorists work together with experimentalists, or does it make more sense to kind of have this balance of first theorists do something and then the experimentalists go bring the topic further and then goes back to theoretical physicists, or what makes more sense?
What makes more sense? What makes more sense is whatever works, right? Whatever brings us quicker to, understanding whatever the hell's going on.
Yeah, so maybe another question for you, Kenny. One of our watchers is interested in your opinion of the CRISPR-modified Chinese babies that were born in 2019 and that we've heard a lot about in newspapers. Yeah,
so this is a pretty difficult, or that was a pretty difficult situation, I think, for science in general, because it kind of showed the danger of this technique again. So it was an experiment that most experts would say was done way too early with way too little knowledge about the side effects, the long-term effects, about everything. There's a clear list of what has to be assured before we can think about editing humans. For example, now in the treatment that I just discussed, there was a huge debate on how to make it sustainable but also reliable, so that we can actually benefit from it and rather don't cause any harm. So all of those things were ignored when the CRISPR babies kind of were edited. And I think so all of these premature experiments, especially when it's concerning humans, are super dangerous, I think, and also harm the outward appearance of science in general, and specifically of CRISPR. So I think,
so that's also the direction of another question, that he wants to know if it's right that if the scissor cut or CRISPR is applied, is not applied in the right place, that cancer could be activated. Can this be stated that easily?
So I think generalizing like that is very difficult, because I mean, cancer has many causes, and there is a factor known about CRISPR-Cas, that it has off targets, meaning that it not only, so it's highly specific but still there are certain sequences that are recognized anyway, even though they are not the actual target. And this is a danger because it's also difficult finding these changes then in the DNA, because you have to sequence the entire genome of the human in order to find these changes. So you have to have a really good target and to perform the genetic changes so that you really can reliably say that nothing else gets changed, no side effects occur. But it will take years and years to understand the entire mechanism behind these off targets in order to rule them out in a way that we reliably can perform these mutations. Causing cancer might also be a side effect. I'm not aware of any study concerning that, actually.
Okay, yeah, the person mentioned a study from 2015, the person asking the question, but yeah, I also don't know. Yeah.
So the audience is getting very curious about black holes, and they're asking questions like, how far away is the nearest black hole, or why are not all stars pushed into black holes? So maybe, Ray, could you give us a little insight on black hole?
Yeah, sure. There are many, many black holes out there. They're just hard to find because they're black, right? But we've never seen a black hole in such detail, at least what happens around it, as the one we've spoken about. There are a few types of black holes. There are the ones that are the size of a few times the mass of our sun, and there's the one in the center of our galaxy, which is 6 million times the mass of our sun. And there's one in, I think it's M87, a different galaxy, which can must be something like 100 times larger than that. They all form in different ways. Most stars, when they die, they won't become black holes. They're not massive enough on that. But the stars above a certain size, above a certain mass, can only, once they finish burning, once they run out of all their fuel, their final end states will be a black hole for a very, very long time. Does that answer the question? I can go
yes.
on for a long time.
Okay, so maybe can black holes disappear, or what happens if they, for example, collapse into each other?
Black holes disappear. Black holes collapse into each other. In fact, when two galaxies merge, the supermassive black holes in their center will eventually spiral in and come into one. And it's possible, in fact, for come into a bigger one. And actually, a Nobel Prize of a few years ago was because when this happens, there are these ripples in space-time that get sent out from the system. And we have these gravitational wave detectors, which now can pick that up. Your first question was about, can black holes disappear? Yeah, black holes can disappear. It's never been observed, of course. It takes a very, very long time. We can't get close enough to black holes to see this process happen. The universe will eventually just be filled largely with remnant black holes, and then what will happen after that, long after we've burnt everything else, the engines of the universe, the stars run out of normal matter to burn, they'll all turn into black holes. But then what will happen is that they will eventually disappear, and they will slowly radiate away through a process called Hawking radiation. At the edge, on the surface of these black holes, there will be a process that is a really beautiful interaction between gravitational theory and quantum mechanics, in which they will slowly radiate away and black hole will lose energy. So yes, they will disappear, and they will disappear with quite a whimper, actually, not much of a bang, a very, very long, slow, drawn-out process.
Thank you so much, Ray. I think you
Sure,
answered
nice question
of the audience. Maybe to close up, would you want to share with us why you chose this topic in physics?
It chose me, and Percentage is a great guy, so it was fun to work with him. Yeah, I guess that's all.
And Kenny, same question for you. What got you interested in your topic?
Yeah, I mean, CRISPR is a really revolutionary technique, and I think working on is super exciting because it's such a fast developing field with always new exciting technologies popping out like in a monthly basis. So it's exciting to work in this fast developing field. So.
And is there anything you'd like the audience to know about your own fields of research, about your own research?
In the sense of what I'm working on? Yeah, so one exciting part is actually that I told you about the viruses infecting those cells, right? And these viruses also evolved defense mechanism in order to cope with those immune systems again. And then again, you have the bacterial cell fighting the fight back. So this is one of the topics then that I'm quite interested in. So yeah, I'm excited about this specifically.
Well, I think we're getting to the end of this event. I want to, first of all, thank both speakers. So thank you, Kenny. Thank you, Ray, for your talks. It was really interesting. I want to thank the audience for asking such interesting questions. And of course, thank REACH for making the event possible and the SIMPACT program possible. Thank you all and have a nice evening.